Assalamualaikum and salam sejahtera everyone. Our group members starting with Syahidatul Badriah binti Baktiah, Anu Najwa binti Muhammad Syafuddin, Mariana Wellington anak Roy, David Gilbert. We will be presenting about thin layer chromatography.
My name is David Gilbert and my student ID is 2021 12 50 47, and I'm going to begin with the introduction of TLC. What is TLC? TLC is a thin layer chromatography. Thin layer chromatography is a technique used to isolate non-volatile mixture. This experiment is conducted on a sheet of aluminum foil, plastic or glass, which is coated with a thin layer of adsorbent material. The material usually used is aluminum oxide, cellulose or silica gel. There are three main components in TLC, which are the TLC plate, the stationary phase, the development solvent, which is a mobile phase, and the sample to be analyzed, which is solute.
My name is Mariana Wellington. For this video, we're using the research paper that we obtained, which is an article of elucidation of chemical interaction between crude drugs using quantitative thin layer chromatography analysis. The objective that we obtained from the research paper is to determine the content of constituent in crude drugs, to understand the chemical interaction between them, and to analyze the target compound in crude drugs according to the HPLC analysis and TLC analysis. The material used is crude drugs and the instrument that used is high performance liquid chromatography, which is HPLC.
Assalamualaikum, my name is Syahidatul and I will explain about the procedure and technique of this research paper use. Firstly, the procedure of this research paper use is 20 grams of cut single cute drug or combination of two mixed drug with a cute drug ratio 1:1 was weighed, and then it is placed in the decoction pot with 400 ml of purified water until the volume was reduced to approximately half of the original volume. And then the extract was filtered and centrifuged at 3000 rpm for 10 minutes, and we got the supernatant. The supernatant was freeze-dried to obtain dry extract. The dry extract were blended in 1:1 ratio of product. And the last one, the extract weight per gram was determined and mixture was used as a blended formula. And the method used in this research paper is extraction method, and the analysis is by HPLC and TLC. Okay, next we'll talk about thin layer chromatography analysis by Oshima et al. In the year of 2022. TLC analysis is conducted in this research paper, and they have 5 products, but I only choose 2, which is phellodendron bark and ginger. This is the extraction method of phellodendron bark. The first one is 0.5 gram of phellodendron bark powder was weighed, and then 30 ml of methanol or diluted hydrochloric acid mixture, which is in ratio 100:1 was added. And then the mixture was heated and in a water bath with the plug cooler for 30 minutes, and the residue was repeated twice with 30 ml and 20 ml of methanol or diluted hydrochloric acid mixture with a ratio of 100:1, and then the last residue 10 ml of methanol was added to the last residue, and it's shaken sufficiently and filtered. Then the filtrates produced were combined and methanol was added to adjust to accurate volume of 100 ml. In this phellodendron bark, we got our targeted compound. This is berberine. For the ginger, this is the extraction method for the ginger. The first one is 30 ml of methanol or water mixture in the ratio of 3:1 was added to 1 gram of ginger powder, and then it was shaken for 20 minutes, centrifuged, and the supernatant produced was separated. To the residue, 30 ml of methanol or water mixture in the ratio of 3:1 was added. Then the steps 1 to 3 was repeated 2 more times, and all the extracts produced were combined and methanol or water mixture in the ratio of 3:1 was added to make accurate volume of 100 ml. Then we got our targeted compound. The targeted compound in ginger is 6-gingerol. In the previous slide, the crude drug was analyzed by TLC, thin layer chromatography. So in this slide, it will analyze by HPLC, high performance liquid chromatography. In this high performance liquid chromatography, it will undergo the instrument, and then from the result obtained, we will get the retention time. For the jingle, which is the target compound is 6 jingerol, we get the retention time 19 minutes, which is longer than other crude drug. For the phellodendron bark, we get 10 minutes for the target compound berberine, which is the shorter retention time than other crude drug. From the result obtained from the HPLC analysis, we can conclude that the ginger has the longer retention time than other crude drug, which means it is strongly bonded to the stationary phase. And then for the phellodendron bark, it has the shorter retention time than other crude drug. This is because it is weakly bonded to the stationary phase, so it will elute first with mobile phase. It will travel faster with mobile phase. For the ginger, it will travel slower with the mobile phase, and then it will elute later than the other crude drug. Okay, this is the targeted compound for this crude drug, which is the phellodendron bark and ginger. For the phellodendron bark, spot 1 to 5 is berberine, their targeted compound, and spot 6 to 8 is their extract. And for the ginger, spot 1 to 5 is gingerol, the targeted compound, and spot 6 to 8 is the extract. For the general thin layer chromatography, I will explain about the detection factor of this chromatography. Firstly, detection factor is the measurement of compound travel, where the distance from the compound original location to the compound location after elution was taken. Detection factor can be calculated by the formula below, which is RF is equal to distance traveled by the compound and the distance traveled by the solvent plant. Then last but not least, this is the retention time. The time that solute or analyte spend in a column or stationary phase depend on the interaction with the stationary phase. So the longer the retention time means that it will travel slower with the mobile phase, and the more it interact with stationary phase, the longer the retention time, and it will elute later.
I'm going to present about the advantages and disadvantages of thin layer chromatography. The advantages of using thin layer chromatography is it was a quick way to separate the components, and it less diverse species of equipment are used in this method. This is because due to the component quick elution, the separation is complete relative quickly. It is possible to visualize all the UV lights component, and these techniques allows the separation of the non-volatile chemical. The thin layer chromatography also separates micro-sized samples. Complex mixes, constituent part, can simply be covered and separated. The disadvantages of thin layer chromatography are it is challenging to replicate the experiment's finding, and it's also applicable to the components of soluble mixture, qualitative research as opposed to quantitative research, and it is not a procedure routine. Thin layer chromatography operates in an open system, therefore, the temperature and the humidity can have an impact on the results. And lastly, due to the limited plate length, the separation process can only go so far.
Assalamualaikum and salam sejahtera. I am Ananaja binti Muhammad Syafuddin. We'll continue the presentation with the concept of thin layer chromatography. Firstly, the sample mixture will dissolve as when the solvent rises up through the silica gel on a plate and will travel up the plate to separate the component. The non-polar compound will move faster with mobile phase because it is weakly bonded to the stationary phase, resulting it to elute first. Besides, the more polar compound will move slowly because it is strongly bonded to the stationary phase, resulting it to elute later. Now, I will talk about the applications of thin layer chromatography. Firstly, TLC is for analyze the ceramide and fatty acid. It also can detect the pesticides and insecticides in food and water. In forensics, the time content of fibers will be examined. Next, it can detect the radiochemical purity in radiopharmaceuticals and can identify the medicinal plant and component. Not to forget, it also can track progressions of reactions. Other than that, TLC can identify the compound present in a given mixture, and lastly, it can determine the purity of a substance. In conclusion, the objective of this study has been achieved as the content of constituents in crude drugs has been determined. The targeted compound also has been identified, which is cisgeneriol, amygdalin, glycyrrhiza, and berberine. That is all from us. Thank you for listening.